Mm. All right, time to get moving and grooving, I guess. Okay, so today what, hope I got everything on. Yeah, looks like it. Cool, good to see some familiar faces too. Anyway, right, so today what we're going to be doing is uh, really just going through some intro stuff, giving you some kind of bigger perspective type things. And part of the reason for that is that it's very easy to get lost in the weeds with genetics. Genetics is not entirely, but in large part about, kind of really small stuff, right? A lot of real abstract concepts. And so it can be really handy at times when you're struggling with these abstract concepts to keep in mind the bigger picture. And I'll do that as much as I can during the classes, obviously, as we talk about different subjects. Um, but sometimes there's just so much information to share that it's a little hard to do that. Make sure I'm recording. Cool, cool, cool. Okay, so what I want to do today is give you a bit of a kind of a broad overview and a bit of a recap of some of the kind of foundational information that you need for this course. And then we'll get on Thursday, we'll start diving into Mendelian genetics, right? So that's going to be our first unit, essentially, for this course. Okay, so let me get up, where are we at? Dang it, where's that gone? It's in there somewhere. I've got so many things open at once. There we go. All right, come on. There we go. Let's get the chat back up. Sorry, I just got to kind of organize my screen. I really wish I had one of those kind of huge, like dual screen kind of fancy setups with like a wood library and stuff. But fortunately, I don't. I'm just sitting on my couch. So screen real estate is a little bit at a premium. I got to fiddle far around. Oh, Doggo's going out for a morning walk. She looks happy. I'll show you. Where you see? There she is, wagging her tail, very happy. She's going to be sniffing lots of sniffs. That's what she does. Okay, so essentially genetics it's a difficult thing to define, right? So I've spent a good number of years trying to define what I am as a scientist. And it's taken me some time to realize that I'm a geneticist, right? And part of the problem with kind of figuring that out is genetics can mean a lot of different things to different people, obviously, right? Depending on their own particular perspective and their own context. So if you talk to me about genetics, about my work, it's going to be a lot of molecular and cellular genetics, right? So how do genes work on, uh, a kind of very small scale, like their structure maybe, particularly their regulation, right? So a lot of what I work on is genetic pathways, right? What works on what, which works on what, and how do these things all work together, right? So for me, genetics is very much a logic puzzle, right? Of sticking together this and this and this. Sorry, I just saw Gabriella's comment. 4 cats is a lot. Not a huge amount, but it's a good amount. I've got 3, but they tend to kind of spread themselves out. Yeah, so I'm really interested in kind of how different things work together, right? Kind of joining dots together. And what effect that has at a cellular level, in terms of metabolism or cell death, and then how that affects kind of things at an organismal level, right? So already just talking about that work, it's pretty obvious there's a lot of different scales involved, right? And that's another kind of common theme amongst geneticists or genetics as a subject. It depends on the scale that we're talking about, right? So molecular genetics tends to be a very small scale, right? Kind of almost what one would call reductionist, right? Kind of breaking things down to their smallest kind of details. Oh, go on, dog. She's acting all funny. But then obviously, genes are in an individual, right? So if you want to study, particular traits, now you start thinking about, well, how those traits spread through from generation to generation, right? How is a particular particular trait. When we talk about trait, we're talking about, like hair color or nose size, or, whether you have hairy toes or not, for example. All right, and so how does that trait spread through generations, right? And if you think about it yourself, I can pretty much guarantee that if I ask you, individually, tell me a trait that is common in your family, right? Think of something which, Grandpa Joe or whoever has that you have, and when you look at those real like old pictures of family, you can identify someone who's in your family, right? So these are things which are unique to kind of your family, and you can actually follow them down through the generations. Right, I can tell that I've got my nose, well, not literally, obviously, but genetically, almost certainly from my maternal grandfather. Now, his family history is a little bit shrouded in mystery for all kinds of reasons I won't go into. So we don't know who he got his from, but certainly it's come through from him through my mother to both my brother and I, right? And I'd have to have a good look at my kids. Thankfully, I don't think either of those got my nose. They got a kind of a mishmash of mine and my wife's, right? So it's a really interesting thing to think about at that level, right? When we talk about, we're talking about individuals and kind of passing things down through generations, right? So again, and if you're kind of often bring genetics back to medicine, not really because I'm failed out MD or something like that, but it's a very easy kind of context, right, to put things into. So if you think about inheritance, well, the classic one is inherited disease, right? So if your parents have and these are kind of typical questions that you'll get in an exam as well. If one or more of the parents has a disease or carries a disease-causing allele, what's the probability that their offspring, one or more of their offspring, will get that disease or carry that allele, right? So when doctors are looking at particular diseases, they often look at family history as well to try and figure out, where's this particular problem coming from? So that's a kind of a different application of genetics. If you talk to my colleague, Dr. Page, who is the one who used to teach genetics, you'll be taking biostatistics, yay, most fun subject ever, with him, most likely later on in your degree. That's kind of what he's teaching now. But he's a population geneticist, right? So he's concerned with not just individuals, but looking at kind of communities, populations of individuals, in particularly lizards and amphibians and kind of like small things with spines. And so he cares about gene flow, right? So how do alleles kind of enter and move through populations, and what kind of things shape that movement, right? One of the things he's interested in is kind of water availability in the hill country, right? And obviously you could think how that would affect lizards and amphibians. And so he's kind of like tying in genetics at a population level with the environment as well. So we start kind of, merging into other areas of biology at the same time. And then obviously if you take that over a long enough period of time, you're talking about evolution, right? So really it kind of the scale that you work at kind of in a sense defines what kind of geneticist you are. But really these things overlap, quite considerably. If you're a medical researcher, obviously you care about genealogies, right? Family histories. What's the mode of inheritance of that particular trait? Because that tells you a lot about what's going on at a molecular level, right? And then you also want to know what is actually causing this problem, right? What change to a gene or genes is, resulting in the particular pathology that you see, right? So really when we talk about genetics, almost certainly anyone who studies genetics doesn't study just one little part of it, right? Because it's a lot of the lines or the names that we give genetics, like molecular genetics or cellular genetics or whatever, right? They're really kind of arbitrarily lines drawn in the sand. They don't really, not say they don't exist in nature, but they an artificial imposition by us in order to kind of better describe what we're doing, I guess, right? And then that's also there's a fairly new brand of genetics or field of genetics, which is called genomics, right? Which we'll talk about a little bit later. Okay, so really when we talk about genetics, a kind of common defining theme is that of heritable material and heritable traits, right? So we're not interested in whether or not you like pop music, right? So I've just watched Trolls World Tour, so that's kind of on the top of my mind right now. Or if, I don't know, politics, which way you prefer to vote, right? Not interested in that. That's very unlikely to be something that is heritable, right? At least in the term which is described. So really we care about traits that are heritable, and essentially those traits must be encoded somehow and to some extent in the genetic information that you receive from your parents, right? Whatever form that may be. And there's more than one form in which you can inherit information from your parents. It's not just nuclear DNA. There's lots of other things as well, right? So, and essentially it's about what underpins that trait, as in what are the genes that contribute to it, right? How do those genes and their forms, right? So their alleles vary within populations and between different species, right? So that's of particular interest. and how are those traits inherited, right? And so really whatever kind of particular brand of genetics you identify best with, it all boils down to that, right? Heritable traits, and how are those traits inherited, and what's the basis for those heritable traits, right? And they might not always be, as I said, nuclear DNA, right? There are traits which are inherited, and they're fairly nebulous and difficult to pin down, which are the result of epigenetic marks in your genome, right? So not DNA based, but actual chemical modifications, right? Which is a pretty trippy thing to think about. Right? These are really what, and these are kind of different names for different things. You ask, get 10 geneticists in a room, and they'll all give you a different name for the same thing, right? So when we're talking about inheritance between individuals in a family or between generations, right? Another way of talking about naming that is calling it transmission genetics. Other people call it Mendelian genetics as well, after the guy that first laid out those principles, right? And that's again another kind of catch can, right? Because there's an awful lot of inheritance which is not Mendelian. In fact, most of it, to be honest. Right. So again, that's just another example of different strokes for different folks. Obviously, molecular genetics I've already talked about, and evolutionary genetics. This is an interesting one because it kind of ties together population genetics, right? Which is how really this should read alleles, not genes, but how different forms of genes change in frequency in a population. And this is the key one, right? Over time, right? So really when we talk about changes happening over time, then we start talking about evolution, right? Don't necessarily talk about speciation, right? You'll get into this more when you actually study evolution later with Dr. Border. Right? She's a far more well, she knows more about this shit than I do, basically, right? Because I'm not much of an evolutionary geneticist. I actually was an evolutionary geneticist for a while, which is kind of weird, right? Because I'm definitely not one. But anyway, but the key is that these are changes over time, right? And if those changes become significant enough to impose barriers in reproduction, then you start talking about speciation, right? So that's kind of I don't know. It's kind of a neat thing to talk about. Also, it's worth pointing out that genetics is a pretty young science. Oh, it kind of is and it isn't. As in terms of a tool, genetics is pretty ancient, right? If you think about it, so my favorite example is teosinte and maize. Actually, let's see if I can bring up a picture. I'm going off reservation temporarily. Teo, see if I actually got this spell right. Come on, images, Google Images is what I want. There we go. Let's get you sharing an actual screen. Doop, doop, doop. And you can see yourselves, which is kind of spooky. All right, so where is it? Here we go. Where's the better pictures? Trying to find some good pictures. Dum, dum, dum. I see this at the side of the road all the time when I'm on my bike. It's kind of it's both a weed and also, let's stick you over here. Also kind of like a grass as well, right? Used as fodder. Anyway, so this is commonly found in Central America. I think it originated in Mexico originally, but obviously kind of spread geographically. Or rather, it may have just spread and then Mexico got defined as being where Mexico is, which is probably more likely. But anyway, this is the ancient ancestor of corn, of maize, right? It doesn't look much like it, right? But essentially over, I think probably I'd have to look into the history in more detail, but at least 1,000 years, right? Farmers in Mesoamerica essentially used principles of genetics, right? What we now know as genetics to selectively breed, and through, a bunch of, kind of accidents, essentially genetic accidents, to generate essentially what we now know as, maize, or, corn on the cob, right? If you want to eat it rather than give it to your animals or make flour out of it. And you can actually see the progression here, right? So these are kind of intermediaries which have been selected for. This is let's get this picture up a little bit bigger so you can see what I'm pointing at. Right, this is the not kernel, because that's the individual little seeds and stuff. Cob, I guess. I don't know what they're called. I'm not really much of a botanist, to be honest.
So,
Sorry?
oh, sorry to interrupt you, sir, but yeah, that could be the cob, the thing that holds the kernel, the center part. It
Right.
could be, I don't know.
Cool. Yeah, I mean, that's as good as name as any, right? And I think we kind of all know what we're talking about when we say that. So anyway, this is the one from Teosinte, and obviously you wouldn't want to be putting that on your dinner plate, right? You wouldn't be able to get an awful lot of butter on it, for example. But over hundreds, if not thousands of years, selective breeding, kind of genetic accidents, and so on have led to something that would be unrecognizable to people who lived, 500 or 600 years ago, right? They'd be kind of astounded, essentially, that we have these, really large cobs from plants which are related to teosinte, right? So really, in many ways, genetics is a ancient field, right? But when we talk about, let's get back to the PowerPoint. When we talk about genetics in a formal sense, right, and you can also think about, breeding animals and, all kinds of other things. Wheat is another great example too. But when we think about genetics as a formal process, right, really it's only been since Mendel, right? And so Mendel was the first one to um, really nail down inheritance, right? And we'll talk about this perhaps on Thursday when we get into it. But there's a lot of arguments around his time about how traits are inherited. A lot of people thought it was called blending, right? So essentially, if you had a tall parent and a short parent, then your children would be, a mix of those two, which actually, in some senses, if you look at your own self, or if you have kids, your kids, if not, then your parents, you actually do see a little bit of that going on in some traits, right? So we're not one or the other, we're a little bit of both. But Mendel was really the first person to lay out the principles of inheritance, right, in a very kind of mathematically sound manner, right, which hadn't been done before. The crazy thing was his work was actually ignored for a good 50 years until the early 1900s, when people went, oh shit, how about that? And started doing Mendelian genetics all over again. It's like, guys, do you not read? I think this was back in the time before internet, so it was kind of hard to find Mendel's paper because it was probably on like a book in some Austrian library somewhere where people didn't go. But anyway, really, stuff didn't start taking off until the 20th century, right? So early 1900s, Thomas Hunt Morgan was really the father of genetics. Mendel's more the grandfather, I guess. Started working on fruit flies, Drosophila, which you'll be working with in lab this semester. They're both cool and really annoying, which is kind of how I tend to think about fruit flies in general. But anyway, he started laying out, essentially fleshing out what Mendel had started, right? But also getting more into the real life complexities of genetics, which Mendel either ignored or kind of conveniently skipped around, one of the two. We're not really sure. And so Thomas Hunt Morgan kind of laid out the idea of chromosomes, right? He was certainly the person that discovered linkage, right? So whenever you talk about genetic distance, you know, how far genes are apart on a chromosome, you'll use the term centimorgans. Comes from Thomas Hunt Morgan. He didn't name it himself because that would be like, kind of like, oh yeah, I'm so crazy. It was his um, scientific offspring, essentially, his PhD student called Stuyvesant, I think his name was, named genetic distance after his mentor, Morgan. So whenever you see like that little c, big M for centimorgans, that's where it comes from, essentially. But the crazy thing was, even then, right? So we knew that DNA was made up of 4 bases, right? We knew that those bases had complementarity, right? So A is only bound with T's, C's with G's. We knew that DNA replication was semi-conservative, right? So we knew all of this stuff, but we still didn't actually know what the structure of DNA was until the middle of the 20th century, right? So really everything in terms of molecular genetics has been in the last 70 years. And really, truly, to be honest, molecular genetics has really been only in the last 40 years within my lifetime, right? And that was from the um, discovery, I guess, the formulation of the process of PCR, polymerase chain reaction, and that changed everything, right? That was probably one of the most transformative discoveries in the history of science, in my opinion. I'm a little bit biased, right? Because that's kind of my personal fave thing. But anyway, so and this is it's kind of fun to talk about this stuff, which is why I'm spending a little bit more time on this than I probably should, because it's just neat to talk about, right? So it's kind of funny to think that a lot of stuff that we know now was not known a little while ago, right? And it kind of sounds obvious when you say it like that, but there's an awful lot of stuff which we didn't know until very recently. So you only have to go back to the 40s, for example, that we didn't know for sure that DNA was actually the heritable material, right? And the reason why we thought that or scientists at the time thought it was most likely not, was that DNA is actually a pretty simple material, right? It's only made up of 4 distinct bits of information: A, C's, T's, and G's, right? And so the question was, how can that simple, very basic structure, right? It's really not much more complicated than, I don't know, sugars, right? Starch, cellulose, things like that in terms of complexity. How can that simple material result in such a huge diversity of amino acids, right? 20 amino acids, and proteins, right? We didn't know it then, but we know now that we have about 20,000 genes, give or take, right? So how can you get that degree of complexity from just 4 bases, right? Obviously, we now know why, and we'll talk about that when we get to the genetic code and things like that. But until these 4 people came along, because there was a bit of a race going on, to be honest. This was really the big prize of mid 20th century science, was discovering the structure of DNA. And so there are a lot of people working on this. There's one particular group that thought it was a triple helix, which would have been super cool. That'd be like Fifth Element, you know, but 50 years beforehand, which is one of my favorite films of all time.
Here's a good one.
Always go, multipass? Anyway, which is nice that some people get that joke. Obviously, people are staring at me blankly as well. Watch the film. It is absolutely cool. I think the costumes were designed by Jean-Paul Gaultier. For a sci-fi film, that is actually pretty cool. Got Gary Oldman in it as well, which is also a good reason. Anyway, complete tangent. But essentially, this lady here, Rosalind Franklin, was a top chemist who was working with X-ray diffraction, right? So X-ray diffraction is a pretty cool technique, which I really don't understand very much at all. Essentially, what you do is you hang on, let's back it up a bit. Light microscopy works for small things, to us at least. But the limit of what you can resolve is actually see, like define an edge with light microscopy is a little bit bigger than the wavelength of light, right? Which is like 4 to 7 nanometers, I think. I think so. I'm not entirely sure on the units. 4 to 7 somethings, like really small somethings. Anyway, when things get smaller than that, you can't see them with light microscopy. So the next thing after that are X-rays, right? And essentially the wavelength of X-rays is that much shorter than the wavelength of even blue light, ultraviolet light. And so you're able to resolve much smaller things. So the idea is you basically make a crystal lattice, right, of whatever it is you want to look at, a protein or DNA or whatever, right? So you have this high ordered repeating structure. You shine an X-ray source through it, and then those X-rays will be diffracted, like they'll be bent. Like when you're kind of looking into water and you're trying to catch a fish, and that fish is not where you think it is, that kind of thing, right? And that pattern, somehow magically, I really don't understand how they do it, tells the people doing that work what that structure looks like. That's where the, I don't know, kind of thing comes in, because I really don't know how they do that. It's a mystery to me. But anyway, this lady here was working for Maurice Wilkins in the UK, lab just outside London. I wish I could remember what it's called. I'd have to look it up. But anyway, she had taken a bunch of pictures of crystal DNA structures, essentially, right? And she went off to a conference, I think it was, and Watson and Crick happened to be visiting Maurice Wilkins at the time, and they, as the story goes, saw those pictures on his desk and went, "That's the answer." And so Watson and Crick got the lion's share of the, I don't know, credit for the discovery, whereas really the actual work in large part was done by Rosalind Franklin, who did not get the credit for the work, which is one of the great shames of genetics, really. And not without precedence. I mean, Marie Curie, for example, didn't initially get the Nobel Prize for the discovery of radium and polonium, right? So, but anyway, and unfortunately, she died before that Nobel Prize was awarded, so she wasn't actually recognized for that work, which is again a terrible shame. Hopefully, we've gone past that a little bit. But essentially, once that kind of kicked out, it led to all kinds of stuff, right? So obviously, you know all this stuff. We've got our anti-parallel double helix. I'll talk about that a little bit more later. But once you start figuring out, okay, we've got a sequence of bases, then you can start figuring out an awful lot more stuff, right? We can start figuring out how DNA gets replicated, right? We can start figuring out how that information encodes proteins, right? Essentially matching up DNA, what we know of DNA, with what we know about the sequence of proteins. Right? And obviously, once you know what the structure of DNA is, you can start figuring out how to copy it as well. And that was really the discovery that, or the process that changed all of modern biology, really, as we know it. And then also how those naturally occurring changes have, for example, caused what was originally teosinte over time to become maize or corn, right? It's not a kind of a magical process. It's really just a simple accumulation of mutations. And then once we started with PCR, then, okay, now we can start thinking about not just 1 or 2 genes or 3 genes or whatever. Now we can think about all the genes of an organism. And really that's the field of genomics. And so that's something which is at most 20 years old. right? The human genome was published in 2001, I think, or 2000, right? So within some of your lifetimes, right? Um, Before some of your lifetimes as well, but pretty recently in kind of in terms of genetics. And things have just exploded from there, right? So we know an awful lot more about where we come from as a species, right? As a result of this, we have a much better, if still a little bit obscure, picture of our kind of ancestry, right? In terms of previous Homo species, or even uh, interbreeding with Neanderthals, right? In ancient Europe, how we came out of Africa as a species and things like that. But also we now have the ability to target specific drugs to specific peoples, right? Because now we understand that, you know, different people respond in different ways depending on their personal genome, right? So if you're lucky enough to have really good health insurance, and you get cancer, one of the first things that will now be done is that you'll have your genome sequenced. And what they'll do is they'll actually look, or they being doctors and geneticists, medical geneticists, they'll look for your genetically encoded sensitivity or resistant to particular chemotherapeutics,
Well,
right? Which varies.
So,
Sorry, someone have a question?
yeah, can you explain that again?
So the easiest way to explain it, and this is one of my favorite things to ask a class, is who here doesn't take ibuprofen if they have pain or a headache because it doesn't work?
I do. I don't.
I
I just
don't.
don't take medications in general.
I
But
Yeah, I tend not to if I can help it, but
I let my body fight everything naturally.
right. Well, I mean, that's really interesting, right? Everybody has a different response to these drugs. For me, most painkillers don't do anything. I actually had to stop my dentist once as he was busy trying to yank a tooth out of my mouth. I was like, hang on a second, this really fucking hurts. I need more of that stuff. He's like, no, you don't. You're just being silly. I'm like, no, really, that just doesn't do anything for me. And I've got other more horrifying examples, which I'm not going to share with you.
You mean he didn't? Oops, sorry, sir.
No,
Sir,
go
you
on.
mean he did? Your dentist didn't numb you or anything like that?
Oh, he
before
did. He gave me the standard dose for, a wisdom tooth extraction. And that, it kind of like took the edge off it, I guess. But it still freaking hurt. And so I was like, I need at least, twice that much for it to actually do anything. And the only time I take ibuprofen, I have to take, yeah, all right, okay. So we're getting the childbirth comparison here. I don't have anything to compare with that. So, actually, no, I'm not going to go into that. But anyway, yeah, that's by all accounts pretty bloody painful too, as one can imagine. But anyway, getting back onto the topic at hand, there's a couple of different reasons for that, right? So all medicines are kind of essentially toxins, right? So they're metabolized by our liver. They're broken down by enzymes in our liver to less toxic metabolites. And some drugs have to be broken down to be active, right? By enzymes in your liver, so that you're given an inert form and they're metabolized to become the active form. And other drugs are active and then they're broken down to inactive metabolites by your liver, right? So I don't know which is the case with ibuprofen, but whatever it is, I have to take at least twice the dose, if not 3 times the dose, for it to actually have the same effect as other people, right? And some of you, it's quite likely that you have to be very careful about taking painkillers because you can actually have adverse effects because you're too sensitive to them, right? So for any particular drug, we each individually have a personal kind of reaction to that drug, right? In terms of how we respond to it. Either we respond to it, very well, we respond to it normally, right? So there's kind of a bell curve potentially here in terms of population level. Look at it. Here you go, population genetics, right? We kind of slip back into that without even thinking about it. Or you might not respond to it at all, right? So when in the olden days, right, when someone had cancer, a particular type of cancer, let's say, they would be given chemotherapeutic, like there's like a list. All right, well, we hit that one first, and then we wait, a couple of months, see if it's having any effect. And if it doesn't, then we'll try that one, and then we'll go for that one, and then we'll go for that one, right? By which time, potentially, the cancer keeps growing, right? So there's this real trade-off. And also, you don't want to give too much because these things are pretty nasty drugs, right? So you don't want to poison the patient and to try and cure them. Whereas now the approach is, well, we're going to sequence your entire genome, and we know we're looking for particular genes which are involved in metabolizing these compounds. And depending on which form of that gene you have, we are able to tell whether you're going to be responsive to this chemotherapeutic or not, right? And also whether you're likely to build up resistance to this chemotherapeutic, because that's a big issue in cancer treatment too, right? And they can basically figure out a large chunk of that, not all of it, but a large chunk of it, by simply looking at your genome. Not simply, but by looking at your genome, right? So we've kind of gone from the kind of stumbling out in the dark kind of thing when it comes to medicine to being able to very specifically target medicine to patients, right?
So I have a question for you.
Uh-huh.
Not to interrupt, but
That's fine.
you're saying they sequence your DNA. If you have cancer, to kind of see what you're susceptible to, could they potentially use the CRISPR machine to essentially rid you of cancer?
Oh, good question.
Because I mean, technically, your DNA is in every cell, so they'd have to.
Right. So, not for all cancers, but for some cancers which are in rapidly proliferating cells whose populations get replaced quite frequently. And I'm thinking really about like leukemia or any bone marrow based cancers. That is actually something that they're trying to do.
Really?
So not just by using CRISPR, there's a couple of slightly older technologies like TALENs and things that they use. And the idea is essentially you take out that person's cells, like bone marrow cells, you genetically modify them to get rid of that error, whatever that may be. And then you reintroduce those cells into the patient and they expand essentially clonally and replace the cells which had that mutation.
That's interesting.
So for some types of cancers, yes. Other types where it's in kind of existing somatic tissue like lung cancer or skin cancer, that's a much, much harder thing to think about. But people are thinking about it for sure.
Yeah, I was just kind of curious about that.
With people that cancer, like doctors, like when they try to like look at you, they use genomics to figure out what you can take.
That's the best standard of care. Not all will do it because it's still quite expensive. It costs, I think, $5,000 or $6,000 to have your genome sequenced, which is kind of astonishing because it cost about $1 billion to do it the first time around, 20 years ago. So the price has come down a lot. So yeah, if it is possible, they will do that, and they'll try and figure out the optimal mix of medicines to kill those cancerous cells and not harm other cells, right? So that they have like the biggest, best shot initially, rather than kind of doing it by trial and error, which was previously the standard of care. Yeah, so it's kind of crazy, and there's all kinds of other immunotherapy is like a big thing. right now in cancer treatment, which I don't really know a huge amount about, but it's kind of like tricking your immune system into attacking your own cancer cells, which is that's like the latest hotness when it comes to cancer therapy. Again, I'm not a cancer specialist by any stretch, but it's a very interesting kind of topic to talk about when we talk about genetics, because it really kind of spans a lot of different facets of genetics, I guess. Okay, so that's kind of like the big picture stuff, and wherever I can, I will kind of pop back in, maybe go off on tangents, talking about how these particular principles can be applied, right? Because again, genetics is a very abstract concept in many ways, right? It's very hard to really see all of what goes on. And really classical Mendelian genetics is kind of the most kind of tangible, I guess, form of genetics. But basically just to kind of ground this material in something that you can grasp, right? It's not just a abstract collection of facts. All right. So we'll see how much of this we get through today, because, you know, I chunner on a little bit sometimes. And, you know, genetics just is very interesting. I mean, it's funny. I often have this argument, going off on a tangent again, with some of my friends at work. I'm like, you know, well, genetics underlies everything, right? At the end of the day, whatever aspect of biology you want to talk about, really, genetics underlies all of it. And then one of the chemistry professors is like, yeah, well, you know, chemistry underlies all biology. I'm like, oh, yeah, I guess it does. And then the physicist jumps in and is like, yeah, well, really, physics underlies all of chemistry. And so you get into like this giant pissing match about, you know, whose is the best science and stuff. There's actually a guy, I think it was a guy called Hardy. It's either a guy called Hardy or the guy who broke the Enigma machines in World War II. Can't remember
Alan
his name.
Turing.
Is that Turing?
Yeah.
Yeah, is it one of those two? But basically, you know, it said the very smartest people are mathematicians. Those who aren't quite as smart do physics. Those who fail at physics do chemistry, and those who aren't good at anything do biology. Always makes me laugh. Anyway, all right, so I'm going to take you through a quick whirlwind tour of some of the basics that we're going to be referencing on. And in many ways, this really leads into, you know, some of the transmission genetics, right? So really, in many ways, genetics is all about information, right? Information on how to do something at a, you know, molecular or cellular level, and how that information gets used and how that information gets passed on, right, is another way of thinking about genetics, right? And so really this is, overall, this is just a, you know, just a kind of an overview PowerPoint. It'll probably take about the rest of the lesson, I think. It shouldn't be too hard to get through. A lot of this stuff you know already. It's really just making sure that, you know, it's at the top of your mind, essentially. So, and this, it's kind of weird. I'm not really much of a PowerPoint kind of teacher. I tend to use, anyone who's had me before, they will end up having to draw out lots of cats on their notebooks. Ask anybody who's had me before, like, oh yeah, cats. We're not going to be doing cats this semester, I'm afraid. Although cat genetics is pretty cool. But anyway, so if some of this appears a little awkward, it's because the format is a little strange for me. But essentially, if you want to talk about genetics, you've got to start with the structure of DNA, right? And so you should all be familiar with this general concept, right? The fact that DNA is made up of nucleotides, right? Or if you want to call them formally, deoxyribonucleotides, hence the DNA part. And each nucleotide consists of a phosphate, a sugar, and a base. These phosphates and sugars, they form the backbone of the DNA strand, right? So it's actually called the phosphate sugar backbone. Apart from people who work on Drosophila, scientists don't have much of an imagination, right? So we tend to just call it as it is. Drosophila scientists are just plain straight up annoying. Anyway, just like one of my little rants. I'll try and avoid going into that. Giant pandas are the other one. Anyway, so this is the information, right? So these are nitrogenous bases, right? They're nitrogenous because they've got a whole bunch of N's in them, nitrogen atoms. And one of the key points of this, going back to kind of GenBio 1 type or GenChem type stuff, is that these are going to be polar covalent bonds, or ability to form polar covalent bonds, right? So, pardon me, anywhere you have an N and an H, or a C and an O, or again an N and an H, so you can look through all of these. These are able to form hydrogen bonds. And actually, if you want to get all kind of like anal about it, if you look at this adenine, right, and so this is where we can get into the weeds a little bit. This has two spots that have polar bonds, right? Because a carbon hydrogen is non-polar, right? It's not different. Only nitrogen and oxygen and fluorine form polar covalent bonds, right? So we've got a amine group here, and we've got a nitrogen in the middle of the ring there. How many bonds can adenine form with thymine? Hydrogen bonds. There's two polar spots in this nitrogenous base that's going to be facing the strand on the other side. How many hydrogen bonds is it going to form? Two, right? Look at guanine or cytosine. We've got an oxygen, we've got an NH, and we've got an amine group, which is essentially a bunch of NHs. How many hydrogen bonds do you think go between guanine and cytosine?
3.
1, 2, 3. Exactly, right? So essentially the very basic structure, and I mean that in a kind of simple sense, not a chemical sense, of these bases is what allows them to bond to each other in a very, very specific way, right? So adenine will not or should not bond with guanine or cytosine, right? Because there's an inherent mismatch both in size, right? Because this has 2 rings, this also has 2 rings, but also in terms of how many hydrogen bonds that base can form, right? So guanines only bond with cytosines because of the number of hydrogen bonds, and also their, 2 matches with 1 essentially, right? So you only get a double ring base, which is a purine, will bond with a single ring base, which is a pyrimidine, right? So, I mean, in many senses, the reason I'm pointing that out is that you can figure this stuff out kind of a priori, right, from first principles, right, if you know what those first principles are. So anyway, essentially, this phosphate sugar backbone is going to be the same, whatever the sequence of the DNA is, right? This is what gives DNA some of its properties, right? So if you kind of like squinch in a little bit, you'll see that these phosphate groups have negatively charged oxygen, well, ions, I guess. Well, anyway, they have a negative charge, right? And this is really important when we get into chromatin formation later on in the course, right? Because that essentially as part of how DNA wraps itself around histones. Another thing which is super, super important is that DNA has directionality. In fact, of the macromolecules, which are, sugars, lipids, proteins, and nucleic acids, right? DNA has directionality. mRNA, therefore, must have directionality, right? Because it's read or it's created in a particular direction. And given that protein is made using mRNA, protein also has directionality, right? So we start kind of putting this together as like a bigger picture of, well, these particular properties have consequences, right? Because they lead on to other things or lead on to other properties, right? The structure of these bases is what underpins the structure, the double helix, essentially. And actually the anti-parallel nature of the double helix too, because you're not going to get, if you have I wish I could do this with my hands, an A and a T, right? They're not going to go on together. They actually have to be upside down relative to each other, right? So if you ever write out, and let's see down here, yeah, they didn't do it, sloppy people, right? So really all of the letters on one of these strands should be upside down, right? If, kind of fully exercise my OCD, because this is an anti-parallel double helix, right? The strands go in different directions, right? And the directionality of these strands, which goes from 5 prime, you see the little red 5 hash mark, that means 5 prime. That's the 5th carbon in that ribose moiety, that sugar in the center. Count it from the oxygen clockwise. DNA and mRNA goes from 5 prime to 3 prime, right? And the 3 prime one is important because that has a hydroxyl group, which is essential for the dehydration reaction to form a phosphodiester bond with the incoming nucleotide, right? So this tells us that not only does DNA have a direction, but DNA can only be made in one direction. You can only add bases to the bottom of this strand because that is the 3 prime end. And when you're synthesizing mRNA, that also is true. You can only add new RNA nucleotides or ribonucleotides to the 3 prime end, right? And so that generates consequences, right? That of much bigger things. In DNA replication, it means that genes have a direction, but because DNA is 2 strands going in opposite directions, you can actually have genes on opposite strands. They go in different directions, right? So you can get kind of build up a great deal more complexity from this. Okay, one quick note to point out. Oh, sorry, I missed wasn't paying attention to. Wow, that would be really cool, Gabriella. Sorry, I'm just kind of got temporarily distracted by the chat. I should have paid more attention to that. I would love to see your family tree of cats, Gabriella. That would be super interesting, particularly if you have pictures of all the different cats, because cat genetics is pretty cool. And Marina asked a question about checking the genome of a child in the womb. In many senses, they do that already with the antenatal tests for Downs. So they don't necessarily check the genome, but they do check the chromosome count of children. And if you wish to, you can actually have further genetic testing done in addition to that, which will look for a variety of congenital birth defects and use that to make a decision about to continue the pregnancy or not. It kind of gets a little bit into A, religion, and B, ethics. So it's a very difficult subject to talk about, to be honest. And, if you take it far enough, you get into eugenics, which is a really difficult subject. Interesting, but difficult subject to talk about. So yeah, there is there's a lot of, we'll talk about that kind of towards the end of the course, some of the consequences of some of the discoveries, right, that we're making. Because really, you know, it's kind of fun to talk about genetics just on its own, but really science exists in the context of society as well, right? So, you know, we're not making these discoveries in isolation, right? They are actually part of the broader society, and that also has consequences too. Anyway, getting back onto things. So you all know this. This is easy stuff. One thing worth pointing out, however, which you may or may not be aware of, is that DNA is not a simple rope twist, right? Made up of two strands, but it's not like twisting two wires together where you have an equal winding. DNA actually has a topography, right? Like kind of like the hill country, not like Corpus Christi, right? Corpus Christi doesn't have any topography whatsoever. So you have essentially what's called the major groove, right? So that's the bigger gap between in the twist, and a minor groove, which is a smaller gap. And so this is actually really important for a lot of proteins that bind DNA, and there are many of them, right? Typically involved in control of gene expression in one form or another. And so many of those will actually recognize a specific structure. I think TRPR, the tryptophan repressor, has a structure which kind of locks into two major grooves, for example, in the TRP operator, which we'll get to later. So this does actually have importance later on when we kind of get to stuff. So one thing worth pointing out, and did I shut that PowerPoint? I've got some pretty pictures for you. Give me a second. Dum, dum, dum. Here we go. It's in here somewhere. I'm not trying to traumatize you. I just actually want to find some nice pictures. Here we go. Yeah, these. These are laser scanning confocal microscope pictures, and confocals produce the most prettiest pictures ever. Oh, cool. Thank you, Gabrielle. I'd like to see that. And so, what this is? Oh, this is a neuron with my. Oh, yeah, it actually says it in the text. I should read what I'm writing. Right, so this is a neuron. You can tell because it has neurites, and it's actually a growing neuron because it has a growth cone, right? So this beautiful green kind of shooting out, those are actin microfilaments, which are how essentially neurons kind of navigate their way to the right spot. If you ever wondered how, you know, the neuron at the base of your spine can grow a dendrite or a axon actually, all the way to a muscle in your toe and get to the right spot. They did it when you're a lot smaller, which helps. But that's one of the ways in which they do so. They actually kind of grow out. It's a very, very spooky looking process. You'll be able to look up pictures of neuron growth cones. Anyway, so this is a neuron. Neurons are really cool cells. These are keratinocytes, which are basically structural skin cells. And they have a very, very different morphology, right? These are very kind of able to move around. These are very static. You see they have kind of real hard junctions with the cells next to them. They form a sheet, right? And they actually have structural properties as a result. These two cell types have exactly the same DNA in them. Exactly, right? There is no difference in the genetic sequence or the information contained by this cell and the information contained by this cell. And so how are these two cells so very, very different, right? And you could say the same when it comes to other types of cells as well.
And
right?
that Hox genes?
Whoa, hey. Wow. Hox genes are, you should take my developmental biology course if I ever get to teach it again. We talk about those a lot. Hox genes are body patterning genes. So essentially, if you I could try and stand up a little bit, I'll show you my, stick it over here a bit more. So essentially, we're divided up, we're bilaterally symmetrical. Sorry, you're getting me off on a little tangent right now.
Must have got cut off.
I guess so.
He's muted. Professor, you're muted.
There we go.
There you go.
There you go. Can you hear me now?
Yes.
Yes, sir.
Yay. Well,
Yeah,
I've got a,
we missed that entire explanation.
oh, well, cool. Means I get to give it to you again. Actually, let's share the screen. There we go.
Wait, I have a question.
Is it going to be a quick one? Because I'm in the middle of a flow here.
Oh yeah, you can just finish and I'll ask you at the end of the class.
Okay. So this is Kevin's question, which is a really, really good one. We're bilaterally symmetrical, right? So we have like an eye on either side of a center line. We've got like 2 arms, 2 legs, things like that. But we're also divided longitudinally from our anterior or our rostral end to our butt, basically our posterior, right? And the reason why you have arms just here and not here, which would be really weird, or like around where your your hips are or something like that, is because very early on in embryonic development, you are divided longitudinally into different developmental segments. And actually, you can see the evidence for that if you just reach behind you and touch your spine, right? Each of those, each of your vertebrae is half of one of those groups. So the top half of one vertebra and the bottom half of the one above it, that is a kind of developmental group. And the identity of those groups is determined by Hox genes.
Okay.
So you have a particular set of Hox genes expressed like, around your head area, right? And it's actually a little more complicated than that because we don't look like this when we're early. We're kind of like scrunched up little like peanut cross-on type things. But then, the Hox genes that denote your cervical vertebra are very different from the Hox genes that denote your thoracic vertebra and so on. It's a fascinating topic.
Yeah.
But anyway, that's an entire course, right? We're not going to be able to get too far into the weeds on that. Otherwise, we just won't talk about anything else. Someone else had a question. I couldn't catch who. Who was that?
Oh, that was me. Okay, so like, how are you gonna ask us these questions on like the tests?
That's a good question. I am not going to test you on my tangents. They're really more for your benefit in getting the bigger picture and understanding of genetics. I wouldn't expect you to give me a rundown of Hox gene theory and the like, because that would just be unfair. The material for the tests is really the PowerPoints and what I talk about as I go through the PowerPoints, right? So almost certainly, let's get back to what we're actually talking about. Where are we at? Yeah, see, no pictures here. It's a whole lot less interesting. Let's move this down a little bit. There we go. So I will be asking a lot of questions about gene expression, not right now, because this is really just an overview of the process. So any questions I ask you about this will be at a fairly superficial level, right? But in I think it's the 4th section on control of gene expression, you'll get asked a lot of questions about how this is controlled, consequences that it might have, things like that. So yeah, if I start going like, oh, that's interesting, and start talking about some not entirely random stuff, that's not something that you need to worry about. Like, is he going to ask me that? I don't know. I mean, don't worry about kind of gleaning through the words that I say. I'm not an oracle. I don't expect you to take like nuggets of wisdom and be able to repeat those. What I really care about in terms of your studying is you need to know the, oh, I'll get to that in a second, Marina. To know the basic facts, you know, what is an allele? What do I mean by genome? You know, work out the probabilities of Mendelian inheritance, things like that. And I want you to understand kind of what we're talking about, right? So, you know, why is it important that gene expression is specific to cell types, right? Why does that matter, essentially, right? So that's really what I'm getting at. The extra stuff is really color to be able to help you understand and appreciate what we're studying, to be honest. But those differences between those two cells are really truly important, right? Because they are the consequence of differences in gene expression. And that's really what makes us the complex organism that we are, is our ability to do that very, very well, right? And so we'll get to that in quite extraordinary depth when we get to control of gene expression in eukaryotes, for example. Yeah, but don't worry like too much about it right now. It's kind of a tricky answer, question to answer in many ways. Marina, your question about the book, you will not be quizzed specifically on stuff in the book if it is not in the lectures. So the book is really an aid and a supplement to what we're talking about now. It's really another source of information, for one, and it's a good one, right? So if there's stuff that we're talking about you don't understand, go look it up in there, and you may get a better explanation than the one I gave you. And B, or two, is a really, really good source of worked problems, right? So those two things are really the main benefits of the book. I'm not going to be looking through the book and going, ha ha ha, there's some obscure fact in some picture somewhere that no one's probably looked at. That's going to be a question. I don't really do stuff like that. Hopefully that helps. Priscilla, I don't really have a huge amount of time to talk about lab stuff during lecture, but I will be talking. There's no, actually very quickly, there's nothing due this Thursday. It should be due next Thursday, next week, right? If it's not, I'll go double check and write myself a note that I haven't got the due date screwed up, because that would be really annoying if I do. But the rest of the stuff about lab, we'll talk about tomorrow and on Thursday, or you'll be talking about today, depending on who you have for lab. So anyway, what we mean about gene expression is really taking the information in that DNA and selectively, and that's the key, using that information under specific contexts. And that could be what cell, right? So that's kind of neuron versus keratinocyte. It could be time, you know, so early versus late development versus adulthood, for example. And it could be, you know, under environmental context. You know, have a couple of beers, your liver is suddenly going to start producing the enzymes needed to detoxify ethanol, right? Because it's bad for you at a cellular level, at least, right? So gene expression has a whole bunch of context associated with it, which turns that information into a very specific outcome, right? And that process, it's often called the central dogma of molecular biology. It's kind of annoying that it is, because this is actually the sequence hypothesis. The central dogma is it doesn't go backwards. That's another rant of mine. So really what we're talking about in terms of gene expression is this is our information up here, DNA, right? Every cell has the same DNA. But the things that do stuff, typically, but not entirely, are proteins. And it's these proteins, which proteins you have and how many of each you have, that is what differs from one cell to the next. That's what defines a cell, essentially, as being a particular cell or another one, right? And so the process by which that occurs is essentially transcription, right? Which is how you produce RNA, whether it's messenger RNA or other associated non-coding RNAs such as rRNA or tRNA. Use some proteins and some more RNA and you get translation, right? So you turn that mRNA, or you don't turn it, you use it to generate your proteins, right? And this whole process we'll be going into in, this is I think in the second section of the course in quite significant detail. But it's good to kind of get this kind of fresh in your head because it's really important stuff to know. And also, here we go. This is actually a pretty cool picture. So this is a piece of DNA. Let's move these over here a little bit. I wish I had a bigger computer screen sometimes. Right, so this is pretty neat because this actually shows, well, it doesn't show the proteins doing so, but it does show the process of producing an RNA transcript of whatever sort that may be, right? And so essentially what you see here is this is the double helix. It gets split apart, and now this kind of faint green strand is the transcript. You can see that this also forms temporarily an antiparallel double helix, right? So this RNA has to be made in a certain direction, right? And that direction is opposite of its template strand.
Is it 3 to 5?
Yeah, so it essentially you have DNA like so, right? So where like my wrist might be the 5' end, or you can do it that way if you wish, it doesn't really matter, right? We're going to split that apart, and if this is a 3' end, then we're going to have our mRNA transcript go in the opposite direction, at least, you know, for the bit that's being copied, right? So this is a really neat image because it really shows that the importance of that directionality, right? And it also shows you that we've got uracils as well, replacing the thymines that were in the DNA sequence. And the thing that I'm going to place this worm in your ear, don't really worry about it right now, right? But this is important for later. The actual coding strand is this one. And so when you make mRNA, or really any RNA, coding or non-coding, you have a coding strand and a template strand. The coding strand is the one that contains the information. The template strand is the one that's used to make the RNA. It's a little bit of a tricky concept to get right off. But one way that you can double check that is the transcript, the mRNA, is going to be a carbon copy of the coding strand, with the sole exception that thymines are changed to uracils, right? And so if you look at this strand over here, we've got a thymine, uracil, adenine, adenine. It doesn't work out perfectly because of the way they've done the picture. Presumably you have a guanine and a guanine, but certainly we have a thymine and a uracil, cytosine, cytosine, thymine, uracil, right? You can see that this transcript is a copy of, hence the word transcript, of the coding strand, the other one. But because of directionality of DNA and mRNA, the new RNA transcript has to get made using the opposite strand. So again, kind of like a funky way of thinking about things. And just in the last few minutes, I'm not going to finish all this up. We'll finish this up the start of next class. When we get into translation, this is what really matters here, right? So remember back when I said that back in the day, they thought that proteins had to be the heritable material because there's so many more amino acids than there were bases, right? Well, there are 20 amino acids, right? And the only way that you can specify 20 different amino acids using only 4 DNA bases is if you group those DNA bases into triplets, right? And this is what this bit down here means, right? So we've got 4 different bases. If you group them into 3s, that gives you 64 words, which is more than enough to encode 20 different amino acids. If you only have words of 2 letters, 4 to the power of 2, you don't have enough different words for all the different amino acids, right? So this genetic code, which we'll talk about again later, really is the way in which we turn a relatively simple piece of information, right? We just got 4. Imagine if you were to try and write something with only 4 letters out of the alphabet, would be kind of the equivalent, right? But using those just 4 letters, if you group them together into words of 3 letters, you're actually able to make 64 different words. And really, this is what you see here, right? All of these different words encode for different proteins. And this code is said to be degenerate, right? And what that means is more than one word. Let's look up here at phenylalanine, for example, right? We've got 2 different codons, I think. Yeah, there's just 2 for phenylalanine. We always have to check. We have 2 different words that encode for phenylalanine, right? And so either of these words will get you a phenylalanine in the amino acid sequence of that protein. And one of the cool things about that is that it essentially gives you a little bit of protection against mutation. So if you look at, let's say, threonine over here, there's an even better one. There's one that has 6. There we go, leucine. That's a better one. For leucine, you can have any letter at the 3rd position, and you still have a leucine. And you can even change for a couple of those the 1st letter in the word, and you'll still have leucine, right? So this gives you a little bit of a buffer against mutation, and we'll talk a bit more about that next class. So I'm kind of out of time right now. I'm sure some of you probably have other things you need to get to. As always, this will get posted up on YouTube in a little bit. If you have any questions about anything that we've covered today, we'll have time to go over them at the beginning of next class on Thursday. Quick.
So the homework, we can like go in and out of it, right? We don't have to stay in it.
I'm pretty sure you can. My caution is because I've never actually done a homework. I've only ever made them, right? So what you see as students is going to be a little bit different from what I know as a professor. But there's nothing in there that should prevent you from doing that. Okay. If any of you do have any problems with homeworks, and this, I've never come across this problem before, so I'm pretty sure it's not going to be one. But if any of you do have any problems with homeworks, just let me know, and I'll help sort them out for you.
Okay.
Another quick note before you disappear is that for those who have lab with me, genetics lab, this week it will be only online, right? So no need to come into campus if you're coming in just for lab. It will just be online this week because I have stuff in the afternoon that I can't get out of, which I think I sent in an announcement. All right. Thanks very
Have
much,
a good one.
everybody.
Thank you, sir.
Thanks for all the awesome questions, and see you on Thursday, if not sooner.